Log on, tune in, find out. Another good idea from Cambridge.
Okay, so today I'll talk about inferring species tree using a set of discordant gene tree lineages, and this is a maximum likelihood approach. Oh. Anyway, it's fine. Okay, so here, well, we have a species tree which is shown on the left-hand side, and we also have a gene tree topology, and which is shown on this box. And I want to emphasize this is about topology. And we are, in general, we allow multiple lineages per species. But let's say we have just one lineages, and starting from the leaves of the species tree. And the coalescent view of how gene trees evolve is you just looking backwards in time, and sometimes the lineages within the same population they can coalesce stochastically. So say when you trace backwards in time, lineages A and B, they just coalesce. And the thing is that it's really, it's a really stochastic procedure. So it depends on the time. If this T1 is very large, it's larger, then there's more chance that A and B will coalesce. And so you can continue to trace backwards in time, and then this will, illustrate how this gene tree topology is obtained in the species tree. So the well-known phenomena is called incomplete lineage sorting, which means in this species tree you can obtain many different gene tree topology. And so let's, for example, this one is this gene tree is different from the topology of a species tree. And to see how this is obtained, you can trace backwards in time, and then here you can see A and B do not coalesce, and within this time T1. And as I said, this is really just a pure chance. It just happened not coalesce, and within time T1. And then when you trace further backwards in time, and B and C coalesce first. And so this gives you different tree topologies. And you can see you can continue to trace backwards in time, and you get this different tree topology. Okay. It's very easy to see that doesn't matter what a gene tree topology is, you can that topology can always arise in any species tree. And but the thing is, each of a gene tree topology will carry different probability of being observed in a particular species tree. And to better quantify this, we can have this notion called gene tree probability. And the gene tree probability is a probability of this observed gene tree topology, TG, given a species tree, TS. This species tree is a full tree containing branch lengths. And this is we use the standard Coalescent model. And obviously, the larger this gene tree probability is, the more likely this topology will be observed in this species tree. So what's the use of gene tree probability? One application I can imagine is that, say, suppose we infer a set of we infer gene trees from the gene sequences from multiple genes, and which are shown here. There are different topology, and we can either define this a simple likelihood function, and we're given this n trees T1, T2, and Tn, and we simply just take a product of each of the gene tree probability of T1 given Ts, and so on, blah blah blah. And here the assumption is that each of the tree Ti is independent of each other, and so that's the underlying assumption. So this allows us to give to find a maximum likelihood estimate of the species tree, which simply maximizes this likelihood function. And then with this definition, we can do all this well-known approach to do local heuristic search to try to find the species tree that maximizes the likelihood function. So here, in this talk, I'll focus on one particular point in this computation, which is exactly how to compute this gene tree probability, which is the probability of given gene tree topology for a given species tree. So an algorithm was given in a paper by Decken and Sauter, which appeared in 2005. And their approach uses this very well-known result in Coalescent theory. Here we define P sub uv of given time t, which is simply the probability of U lineages coalesce into V lineages within time t. So look at this species tree branch on the left-hand side. We have U lineages and on the bottom, and then we have V lineages on the top. So we can see, with the standard Coalescent model, then the U lineages will coalesce into V lineages. We'll trace backwards in time within time t. And the nice thing about this probability is that it has a nice closed form formula. It looks complex, but for computer scientists like me, it's nothing. Right? It's very easy to compute. And so this is a very good thing, and it has this simple closed form formula allows us to compute this. So this closed form formula for this P sub UVT allows us to compute the gene tree probability, which is which you can see from here. We assume that Coalescent events along different branches are independent, so we can treat the different species tree branch independently. So let's look at one example here, and for now let's assume we know the Coalescent history. Coalescent history, I think Luai stated on Tuesday, which simply tells you for each Coalescent events where they occur on the species tree. So for example, this tree embedded in the species tree will tells you exactly where this along which species tree branch this Coalescent event occurred. And for this given Coalescent history H, we can easily compute this gene tree probability, which is simply as this. So we just look at how many Coalescent events occur along different species tree branch. So in this case, we have a branch of T1, and you can see this is we have two gene lineages, and they coalesce into one lineages. So that's why we have this the probability we multiply a term called P21 of T1. Two lineages coalescing to become one lineages within time T1, and then you can that's like a P21. And you can look at another gene tree branch. Species tree branch, which length of T2. And here you can see that we have two lineages, and they remain two lineages within time T2. So you multiply a factor with P22 of T2. So two lineages remain two lineages within time T2, and you multiply this term. And you continue, and then you have reached the root of the species tree. Now you have an infinite amount of time to make them coalesce. So this will become P31 within time infinity. And you also need to multiply a combinatorial factor. I don't have time to describe that. The main problem for the Decken-Sauter's algorithm is that it's not scalable. Its reason is that there are just so many possible Coalescent history for a given species tree and a given gene tree topology. And for example, the Coalescent event may occur this way, and they can also occur in many different ways. So, that's why the Decken-Sauter's approach which basically just enumerate all possible Coalescent histories, and I'm going to show in a table if I have time later, and it's not scalable. All right. So how to speed up? And this is this is the work I got started. And I'm not going to enumerate all possible Coalescent histories. Instead, I'm going to use something in computer science called dynamic programming. And the key notion here is called ancestral configurations, and which is specified for a given position in the species tree, the set of gene lineages alive at that point of time in the species tree. All right. So let's look at what's the possible ancestral configurations that is on each positions in the species tree. So let's look at the bottom of the species tree. And here, at this particular time, we only have one lineage, so we only have one choice. It's just lineage A. Here we say write on the speciation of a A and B species, and we have this A and B lineage, and so this is the only choice. They don't have time to coalesce yet because they just meet, and they have no time to coalesce. And but since change a little bit, if you look at this point of time on the species tree, and here we have two choices. We have A, B, C, and EC. The reason we have EC is because the A and B may coalesce into lineage E within time of this T1, right? So we have two choices. And when we look at the root of the species tree, we have three choices. And here we have A, B, C, D, E, C, D, and F, D. And that all depends on how many numbers of coalescent events will occur along this branches. And so these are these lineages, a set of lineages are shown here, correspond to the ancestral configurations I'm talking about here, right? So the key thing to compute the gene tree probability is to define this probability of ancestral configurations, and which is basically the probability of the observing the gene lineages specified by this ancestral configurations at that moment in time in the species tree, right? So let me show you how this ancestral configuration probability can help us to compute the gene tree probability. Here we have I'm going to show you a recurrence for the ancestral configurations probability, which allows you to compute the gene tree probability. So here we have this. Well, I'll only show you one place in the species tree. We have time t, and this is the root of a gene tree. On one side we have lineage A, B, C. On another side we have D. All right, and this is and here the ancestral configuration ACs and to this point of time, and the D and the leaf D only have single lineage D, right? And on the other side we have two choices as showed. We have A, B, C, or EC, all right? And the root we have three choices, and for each of the choices a different way of arising. So let's look at the ECD. Particular point ECD. So this means that this ECD is obtained by merging of lineages from left and right, and the lineages coming from left got to be EC. So this coming from EC, right? Lineage on the right will be D. So these two meet and merge become AC of ECD, and then we need to know in which scenario we're going to observe EC right below this speciation event of the root. And they have actually two choices. One is that the A, B, C will coalesce into lineage EC, all right? So in other words, A and B will coalesce into E. And we have another choice, which is EC. EC is that EC will remain EC with uncoalesced along this time of T2. And I'm going to define this transition probability between the ancestral configurations, which this probability can be computed by the previous slides I show you. Yeah. And then this allows us to compute the probability of EC prime, which is simply just summation of these two terms, the probability of A, B, C times the transition probability between A, B, C to EC within time t. And plus the probability of EC, times the transition probability of EC become EC within time of t. And so this allows us to compute the ancestral configurations. All right. So with this definition, it allows us to come up with a peeling style algorithm for computing the gene tree probability, which is basically starting from the root. Starting from leaves. And here, on the leaves of the species tree, the probability of this ancestral configurations are all just 1. And then when we moving back, the probability of AC just a probability of A times the probability of B lineage B, which is still just 1. And here on the divergence, we can see the probability will become again coming from the whatever ancestral configuration coming on the its descendants branches, and this allows us using recursion allows us to compute these terms. And same thing on the root. On root, we need to multiply this probability of EC and times the probability of D. All right. So why this is better than the Degnan-Salter's algorithm? And I want to compare the number of ACs, ancestral configurations used in my method versus that using Beckman-Salter's approach. And here we assume the gene tree and the species tree has the exact same topology. And in one example here, which is a so-called maximum symmetric tree, which is just like caterpillar tree. And here you can see the number of unsuch configurations is just n times n plus 1 over 2, which is a polynomial in terms of number of species. N is number of species. And the number of histories, the Coalescent histories, on the other hand, is exponential. So it's Catanese number. And if you look at this table here, and you can see the clear difference. And with 20 taxa, and you can see the number of ACs for a symmetric tree is just 210. And the number of histories, Coalescent history, is over 1 billion. All right? So I don't have time to describe how does it perform in foreign species tree. And this slide basically shows you my method can be justified. And I don't have time to say that. And that's all.
But okay, we can take it for questions. Any questions? Well, I have one actually. Oh, okay, good.
It's not really a question since he has time. Can you show us the performance in the slide?
The result?
Yeah.
Okay, so I compared the method with a program called STEM. And I tried it different ways. And one way is I just use a perfect the gene trees, which is just coming right out of the simulation, and those results are shown on the dash line, and you can see from here, and the dash line are results for and also this one are results for the perfect gene trees, and you can see this is from program STEM, and when given perfect gene trees, they really converge nicely to the perfect the true result. In that case, my method doesn't perform as well. It also converging, but a much slower pace. Yes. Just a moment. And for the solid line are the ones coming from inferred gene trees. You simulate sequences on the gene trees, then you infer with maximum likelihood, and then those are the two situations you may observe. In that case, this is my method, and that a little bit worse than the one for the perfect gene trees, but not too far off. And but STEM, can quite difference, much larger difference.
I thought your work was about speeding up the calculation of the probability.
Yeah,
which explains the difference in speed. But where is the gain of accuracy coming from? Because it's just a different way of computing the same.